Okay, let's finish our non-specific immunity by talking about the second line of defense, where I think we have 4 or 5 different things to talk about. The first are the white blood cells we've already talked about. As you remember, there were 5 of them. And remember, I said you needed to know functions for each of them. And so notice that I put lymphocytes in red here, and that's because lymphocytes are going to be involved with specific immunity, whereas the other 4 types of cells are involved with non-specific immunity. And so they all get to do their normal jobs in this type of immunity. So let's talk about where they can be involved. And the first process is inflammation. So when we have an inflammatory process, the damaged cells as well as mast cells can release chemical mediators. Do you remember what mast cells were? What type of white blood cell, when it leaves the bloodstream and goes into tissue, do we call a mast cell? Here's a hint. It's because it contains histamine. Okay, so go back and figure that out. Okay, and in inflammation, we have 2 different white blood cells, neutrophils early in the process, and a different one later on, which are the ones that when they move into tissue, we always call macrophages. Those are going to be doing phagocytosis of the pathogen. So go back and review. And before you continue, make sure you understand that. All right. So then we're going to have the 5 cardinal signs of inflammation. And so, yes, I know people say there's 4 cardinal signs of inflammation. That's because there's 4 signs related to the capillary changes. Redness, heat, swelling, and pain, they're all related to the changes that have to do with what happens when we have changes in the capillaries due to the damaged cells and the chemical mediators. And then those things will result in loss of function. If you have swelling and pain in the area, then you're going to lose function. Okay. And all of these inflammatory signs are trying to protect your body. They're telling you, look, I'm damaged. Don't use me. Okay. Give me a chance to heal. So let's go through the inflammatory response step by step so you can see how all these cardinal signs develop, because you need to know exactly how they all occur. So we're going to start in this case with a pin poking on the skin surface, and the pin is bringing some bacteria with it. And as you can imagine, it's not sitting there going between cells. It's actually damaging some cells and causing some tissue damage. And when it damages cells, it releases chemicals as well. So both the injured tissues as well as mast cells that were there are going to be releasing histamine. And what histamine is going to do is it is going to cause local capillaries to dilate. So we're going to have vasodilation. The diameter is going to get bigger. So we can have more blood flow into the area. So when we have more blood flow into the area, that is going to give us redness as well as heat into the area. Now, the other thing is because the capillary has dilated, the epithelial cells are going to be pulling apart slightly, as you can see in this diagram. See how the cells are pulled apart slightly? So that allows the fluid, the plasma, to leave the blood vessel and go into the tissue, which is going to cause localized swelling. The other thing it's going to allow is for those white blood cells to squeeze between, that process known as diapedesis. So, it's bold once again. So if you don't know that term, make sure you do know it. And both of these processes, the fluid and all the extra white blood cells in the tissue, are going to give us that third cardinal sign of swelling. Um, uh, so if that pin had damaged a blood vessel, then platelets would come in and do a platelet plug and be followed by coagulation cascade. So you'd have the whole clotting process going on. Now, you would have phagocytosis at first being done by the first responders, which would be the neutrophils, and then later on, the monocytes, which are the cells that become the macrophages, they would do that starting about day 3 or day 5 and continue on. The normal inflammatory response in a person with a robust immune system will take you 7 to 10 days. So don't expect it to go away in 2 or 3 days. The peak inflammation is going to be at around 48 hours. Also, did you notice I didn't talk about pain? Yes, that's because pain comes from a couple of different sources. We talked about how we could injure cells and we could release substance P, which is a cytokine. Macrophages can also release cytokines, other ones we didn't talk about. And these can all act on those free nerve endings in the dermis there, which would stimulate the pain receptors, the nociceptors. All right. Going on to our third topic, let's talk about another plasma protein system, this one, the complement system. We already talked about a plasma protein system before, and that's when we did coagulation cascade, where we had all those factors which floated around in an inactive form. And then once one was activated, it activated the next one. Well, that's the same thing that happens here in complement. We have their inactive forms floating around in the plasma, these proteins. And the reason why it's called complement, and notice this is complement spelled with an E as opposed to complement with an I. Complement with an I is when I say, oh, you're so pretty. But complement with an E is when it means in addition to other stuff. So this complements the other immune responses that we are having. And so here's what the complement system looks like. In other words, there's multiple ways of activating it. And we don't need to know how you activate it. Some pathogens activate it themselves, like some bacteria walls. Sometimes antibodies, antigens can activate it. We don't need to know how. But anyway, we're going to activate it. And so we have different proteins, and we have different proteins that can do different things in this pathway. So for instance, over here, this part, when it becomes activated, can bind to mast cells. So when this becomes activated, it binds to mast cells, which causes release of histamine, which then is going to give you a more robust inflammatory response. Whereas on this side, if this over here becomes activated, it attracts phagocytes. So you get more phagocytosis in the area. But no matter how it gets activated, eventually everything gets down to this final common pathway down here, leading to this thing MAC, which stands for the formation of a membrane attack complex, which you need to know about. So plasma protein systems do 3 big things: increases inflammation, increases phagocytosis, but forms a membrane attack complex. So here's the membrane attack complex. These are like complements, and you don't need to know. It's like 5B, 6, 7, 8, 9. And basically, it's going into the plasma membrane of the pathogen, and it is basically making a whole circle. And once it's made a whole circle, it basically has punched a hole in the membrane of the pathogen. And so once you have a hole in it, see it sitting there? And this pathogen has lots of holes in it. And what's going to happen is extracellular fluid is going to be entering that pathogen. And basically, that pathogen is just going to start getting larger and larger, just like you were blowing up a balloon until it bursts. And so then this cell has died. And so if complement system is still activating, it's going to be doing this to all the pathogens it can find. Now, the next and final thing that is non-specific is activating interferon. And so this is going to be for viruses, and this is really cool. So watch what I am saying. Here's a virus. So as you know, it is a nucleic acid inside a protein coat, and it needs to break and enter into a cell, which is done here. And then here's the nucleic acid. And so what happens is it incorporates itself as part as the host cell DNA inside you. And when it has done that, it is turning on genes to make lots more of that virus because, as I said before, it's narcissistic, but at the same time, it has turned on genes to make interferons. Okay. So at the end, it is going to be releasing a bunch of interferon genes, but that cell was killed by the virus. Okay. So even though the cell made interferons, it could not be saved. And it did release those thousands of copies of the new viruses, which were released in the wild to now infect thousands of other cells. But here's the interferon proteins, and they are going to bind to receptors on a different cell. And when they bind to receptors on a different cell, they stimulate genes which turn on antiviral genes. And so when a virus comes to this second cell that has already bound to interferon, the antiviral proteins will block that virus from reproducing. Now, if that virus is already there before the interferon blocks, it can't do anything, but the interferon is there first before the virus gets there. Then that second cell is protected against the virus by the interferon. So, in summary, interferon is produced by a cell that was infected by a virus and can do nothing to help that cell. That cell will die. Interferon binds to the receptor of a non-infected cell and can prevent viral replication in that non-infected cell. And because of that, we can take interferons from people who have had a viral infection with something, say hepatitis C. We can take the interferon out of that blood and use it for treatment in somebody who has an acute hepatitis C infection to try to prevent their liver cells from being destroyed by the hepatitis C. So that's where interferon is good. Oh, and the last thing I want to talk about is a fever. A fever is defined by a core temperature of 100 degrees or higher. So I know you think 98.6 is your temperature, but you know your temperature has a diurnal variation as well. So it's got to be over 100 degrees to be a fever. The 101 if you're doing rectal temperatures. Okay. And the reason you have a fever is because your hypothalamus has been reset. Either bacteria has released an endotoxin that has reset it, or your body is doing an immune response where macrophages or other cells are releasing these chemicals called interleukins, which I haven't talked about yet, but I will, which reset your hypothalamus. Okay. So, yes, your hypothalamus is going to reset it and it's going to give you a fever and also causes vasoconstriction to make your fingers and your toes feel cold at the same time, and also activates your muscles, which gives you shivering at the same time. So you have a fever, but you want a blanket and you're shivering all at the same time. So what should you do? Should you treat your fever? Not if you can avoid doing it. Definitely not at 100 degrees. So really try to avoid treating your fever until it's over 102 if you can. Why is that? Because it will slow bacterial growth. Now, you don't have to go all the way up to 104 degrees because at 104 degrees, you can start getting brain damage. So, after your fever is over 102, yes, you can bring it down to make you feel better. But a low grade fever between 100 and 102 actually will increase your immune response. It increases your phagocytosis. It increases your inflammatory response. It increases all of these non-specific as well as some specific responses we haven't talked about yet. So before I sign off, I would like you to take this picture where I have blocked out all of the words and I put a whole bunch of terms that you should know and be able to use. Talk yourself through the inflammatory response and make sure you can use all of these terms when you're talking yourself through this. And thank you so much for all your hard work. And that is it for the first day of the lymphatic and immune system. And I will see you for day 2 when we talk about specific immune responses as well as active immunity.